Hello, my name is Daisy Leung, and I'm an associate professor in the Department of Medicine at Washington University School of Medicine. And today I'd like to share with you some of the work that we're doing towards understanding how the non-structural proteins from respiratory syncytial virus facilitates host immune evasion and how our work can provide a basis to target these proteins therapeutically. So respiratory syncytial virus, or RSV, is a human pathogen. It's a leading cause of lower respiratory tract infections, primarily in the very young, the elderly, and the immune-compromised. It poses a serious public health threat with economic impact. RSV, like most other respiratory viruses, circulates seasonally, and most RSV infections tend to peak in the winter months in the US. However, the COVID pandemic shifted transmission rates, and we started to see RSV infections during the spring and summer months, as seen here on these graphs. And these numbers are likely to be underreported. Currently, there is a lack of an effective and licensed therapeutic or vaccine available for RSV infections outside of palivizumab, which is a humanized monoclonal antibody against fusion protein that is given prophylactically to infants at high risk. However, it is cost prohibitive. However, there are several new RSV interventions that are currently in development, and these are falling in the categories of monoclonal antibodies that are targeting entry, vaccines, as well as antiviral treatments that are targeting entry as well as the viral polymerase. Some of these are summarized here on this table that are in different stages of clinical trials. However, the fact that we still lack a vaccine despite early attempts to develop one in the 1960s suggests that we need to improve our understanding of the viral components as well as the host responses that control infection. So RSV is a non-segmented negative strand RNA virus. Its viral genome encodes for 10 open reading frames, 11 proteins. And what I'd like to highlight here are the non-structural genes, NS1 and NS2, that are shown here in this red box. So NS1 and NS2 are fairly small proteins, about 15 and 13 kilodaltons respectively, and they're found only in RSV and lack counterparts in any other members in this group, including the closely related human metapneumovirus. It is thought to be produced in high abundance due to its proximal location to the promoter. It doesn't have significant sequence homology to each other or any other known protein, suggesting that they are unique. They're also not thought to be packaged as part of the mature virion, hence their designation as non-structural proteins. Early studies by several groups have suggested that the non-structural proteins function as interferon antagonists. Recombinant viruses, where either the NS1 gene or the NS2 genes have been deleted, results in an increase in interferon beta mRNA expression levels when infected in airway epithelial cells. And when both NS1 and NS2 genes are deleted, this results in a significant increase in interferon beta mRNA expression levels, suggesting that NS1 and NS2 may function cooperatively to inhibit interferon beta production. So shown here is a highly simplified schematic of the interferon beta signaling pathway. And just to briefly review, viral RNAs are a pathogen-associated molecular pattern that can be detected by cytosolic pattern recognition receptors, including RIG-I and MDA5. An activation of these receptors leads to the activation of a number of downstream signaling molecules, including the transcription factors BRF3 and 7. And these can then translocate into the nucleus where it can stimulate interferon beta production. And interferon beta can act in an autocrine or paracrine fashion, bind to its receptor IFNAR1-2, and stimulate JAK-STAT signaling. And this in turn can activate the transcription of antiviral genes, leading to an antiviral state. And studies in the last 15 to 20 years have suggested that NS1 and NS2 are important for host immune evasion and that they target multiple points in the interferon signaling pathway, particularly in the cytoplasm, and some of which that are highlighted here. However, the non-structural proteins have remained enigmatic in the sense that we still don't understand how they mediate this function. And so for the rest of my talk, I'm going to be primarily focusing on the left side of the pathway and tell you two brief stories that look at the structural basis and the molecular mechanisms by which NS1 and NS2 inhibit interferon signaling. In the first part, I'll show how we think NS2 functions in the cytoplasm to prevent the activation of the RIG-I-like receptors. And in the second part of my talk, I'll describe how NS1 also functions in the nucleus to modulate host gene transcription. So to gain more insight into the molecular basis for how the non-structural proteins can mediate host immune evasion, we focus some of our initial efforts on these biochemical and structural characterization of NS2. So Sharupa Chatterjee, a former postdoc in my lab, was able to recombinantly express and purify NS2, which is shown on the SDS page. Analysis of NS2 using size exclusion chromatography coupled to multi-angle light scattering revealed that NS2 elutes in a fairly narrow peak with a molecular weight of about 14 kilodaltons, suggesting that it behaves as a monomer in solution. Now, using this material, she was able to generate protein crystals, and we were able to solve the X-ray crystal structure of NS2, which is shown here. So NS2 structure is comprised of a mixed alpha-beta fold. It contains 4 N-terminal alpha helices that is flanked by anti-parallel beta sheet. In comparison of our NS2 structure with all the structures that were in the protein databank at the time did not reveal any significant similarities to any other proteins, suggesting that NS2 adopts a unique fold. So to gain more insight into NS2 function, we look back at some of the earlier studies which suggested that NS2 inhibits host signaling early on in the interferon signaling pathway. So RIG-I and MDA5 are pattern recognition receptors. They contain at their N-terminus 2 tandem caspase activation and recruitment domains or CARD domains. And these are protein interaction modules that interact with other CARD-containing proteins, including the downstream molecule MABs. And this is followed by a helicase domain and a C-terminal domain. So in order to further probe the interaction between NS2 and the pattern recognition receptors, Jingjing Pei, a former postdoc in my lab, performed a series of flag co-immunoprecipitation assays. And what she found was that RIG-I was able to co-immunoprecipitate NS2, as was shown previously. She also found that MDA5 co-immunoprecipitates NS2. And because both RIG-I and MDA5 undergo conformational changes upon binding of our RNAs, we repeated this experiment and treated the cells with poly-IC. And she found that both RIG-I and MDA5 were still able to co-immunoprecipitate NS2. To further narrow down the region that was important for this interaction, she next tested binding of the N-terminal CARD domains with NS2. And so shown here are the results of the co-immunoprecipitation, showing that both RIG-I CARDs as well as MDA5 CARDs can co-immunoprecipitate NS2, whereas the helicase and C-terminal domains do not. Now, because both RIG-I and MDA5, upon their full activation, can bind and activate the downstream molecule MABs, we next wanted to test the impact of NS2 on this interaction. And so shown here are the results, showing that upon increasing concentrations of NS2, the RIG-I and MDA5 lose the ability to bind to MABs, suggesting that NS2 binding prevents the interaction between RIG-I or MD5 with MABs. Now, in order to figure out whether the interaction between RIG-I and NS2 is direct, we next performed in vitro pull-down assays using purified proteins. So shown here on these gels, we have immobilized purified RIG-I cards onto resin through a tag and incubated with purified NS2. And after subsequent washes, we find that NS2 is still retained on the resin along with RIG-I cards, suggesting that the binding is in fact direct. We next wanted to map the binding interface between NS2 and RIG-I cards. So we performed hydrogen deuterium exchange coupled to mass spectrometry, or HDX-MS. And this was work that was done in collaboration with Nicole Wagner and Mike Gross. And just very briefly in these studies, hydrogen deuterium exchange is initiated when the protein is incubated in buffer D2O. At various time points, the reaction is quenched, the protein is digested, and incorporation of deuterium is monitored by the isotopic mass envelope through LC-MS-MS, and we can plot the percent of deuterium uptake in a kinetic plot shown on the bottom left of the slide. And this experiment can then be repeated now in the presence of a binding ligand. And regions that are protected from deuterium uptake due to protein-protein interactions would cause a shift in the isotopic mass, and we will also see a decrease in the kinetic rates of deuterium uptake. So shown on the top left hand corner is the results for HDX for NS2 alone. So shown is the percent deuterium uptake across the molecule as a function of time. And what we can see here is that there are regions in NS2 that are highly protected from exchange, and they're reflected by the cooler colors on the heat map. Regions that are more dynamic and more prone to exchange are reflected by the warmer colors. And these correspond to both the N and C termini of NS2. So Nicole was able to then repeat this experiment now in the presence of RIG-I cards, and we were able to identify regions that were important for the interaction with NS2, and this is reflected in the select kinetic plots that are shown in the top right-hand part of the slide, where NS2 alone is in black and NS2 with RIG-I cards are shown in red. And we can map this results back onto our structure of NS2, and we find that this corresponds to the N-terminus of NS2. And so to validate these interactions and these results, we next introduced several mutations in NS2 at the NS2 N-terminus and tested its ability to bind RIG-I as well as to inhibit interferon beta promoter activity. So shown on the left-hand side are the results of a co-immunoprecipitation showing that full-length RIG-I can co-immunoprecipitate wild-type NS2, whereas we lose the interaction with NS2 upon introduction of mutations at the N-terminus of NS2. On the right-hand side, we find that introduction of different combinations of mutations in the N-terminus of NS2 leads to the loss of suppression of interferon beta mRNA levels. So just to summarize the first part of my talk, I've shown that we were able to solve the X-ray crystal structure of NS2, which revealed that it adopts a fold that is different from other known interferon antagonists. RSV NS2 also inhibits signaling by preventing the full activation of RIG-I and MDA5 and preventing its binding to the downstream molecule MABs. And because previous studies have also suggested that NS2 can interact with NS1, we are now looking at additional studies to see how NS1 binding can modulate NS2 function. So now I'm going to switch gears and now talk about some of the studies we've done in order to identify how NS1 can also facilitate host immune evasion by also having a functional role in the nucleus and modulating host transcription. And much as we've done with NS2, we took a very similar approach with NS1. Here we are able to solve the X-ray crystal structure of NS1, which revealed that is comprised of two anti-parallel beta sheets that are arranged orthogonal to one another and forming a beta sandwich that is decorated by 3 alpha helices. A comparison of our structure of NS1 to our structure of NS2 did not reveal any significant similarities. NS1 and NS2, though, share 4 common residues at the C terminus, and this is D, L, and P. When we looked back at our structure of NS2, we find that the terminal proline residue forms pi stacking interactions with the histidine 37 residue that is present at the end of alpha helix 2 of NS2. And this forms end capping interactions that stabilize the helix and the conformation of NS2. When we look back at our structure of NS1, we did not observe any end capping interactions in NS1. However, the fact that NS1 also contains this DLMP motif it raises the possibility that perhaps NS1 can adopt additional structures depending on different conditions. When we compared our structure of NS1 to all of the other structures in the protein data bank at the time using the DALI server, it did identify several hits, and these included other viral matrix proteins, including RSV matrix protein. And this was a little bit surprising to us given the lack of sequence similarity between the two proteins. However, when we performed an alignment of the two structures with NS1 shown here in magenta and the N-terminal domain of RSV matrix protein shown here in gray, we see that there is remarkable alignment between the two structures. Now, we don't quite understand why there appears to be duplication of a fold that already exists in matrix proteins. However, we're tempted to speculate that NS1 is a structural paralog of RSV matrix protein that rapidly diverge in sequence and in function. Now, while there are many similarities between NS1 and the N-terminal domain of matrix proteins, we next wanted to see if we can identify regions in NS1 that were specific and important for mediating NS1 function. And what we noticed from our structure is that NS1 contained at the C terminus this alpha helix. So we next wondered whether this helix was important for its stability or its function. So in order to test that, we next introduced a series of mutations in the C-terminal helix of NS1. And what we found is that mutation of this helix did not impact its overall stability using circular dichroism. Now, because NS1 functions as an interferon antagonist, we next wanted to test its ability to inhibit interferon beta promoter activity using a luciferase acid. And this work was done in collaboration with Priya Luthra and Chris Bassler. And what we found was that wild-type NS1 was able to inhibit interferon beta promoter activity to levels comparable to that of a potent interferon antagonist, Ebola VP35. We found that mutations in the C-terminal helix led to a loss of suppression of interferon beta promoter activity, whereas mutations outside of this helix had no impact. So these results suggest that the C-terminal helix of NS1 is important for interactions and for its function. So we next wanted to establish a structural basis for this helix in viral replication. And so we next generated mutant NS1 recombinant viruses in collaboration with Mike Holtzman and Marty Moore. And what we found was that these mutant NS1 recombinant viruses were able to form syncytia very similar to that of wild-type RSV in vero cells as shown on the top panel of the images. We also found that our recombinant NS1 mutant viruses were able to replicate at levels comparable to wild type in vero cells, which are interferon deficient. However, we did observe a 2-log difference in viral replication rates in A549 cells, which are interferon competent, suggesting that these viruses are attenuated and that the attenuation are due to mutations in the C-terminal helix. Analysis of differential gene expression showed that on the heat map on the left, that upon wild type RSV infection, there's a significant upregulation of a number of genes. And comparison with our mutant NS1 recombinant viruses along with MOC are significantly different. And what we found is that the genes that were upregulated upon wild type RSV infection tend to fall in the interferon induction, interferon response, and the antioxidative response pathways. So overall, this data suggested to us that perhaps NS1 may have function as a global regulator of host gene expression. So one of the first clues that we had that NS1 had a functional role in the nucleus came from a proteomic studies that we performed in collaboration with Andreas Pielmeyer. So shown here on the left-hand side is a volcano plot of the results from an affinity purification mass spec study, showing highly clustered and significant interactors of NS1. And what we were able to identify were several components of mediator complex. A mediator complex is a co-transcriptional activator of RNA polymerase II, which binds to the promoters of many antiviral genes. And what I don't show here is that the same components do not interact with NS2. And so the second bit of evidence came from the fact that NS1 can partially inhibit interferon beta promoter activity when stimulated by constitutively active and nuclear localized IRF3. So the data here combined suggested to us that NS1 had a functional role in the nucleus. So in order to visualize NS1 in the nucleus, we next infected human tracheal bronchial epithelial cells in airway liquid interface cultures with RSV, and we stained for NS1, shown in green, acetylated alpha-tubulin, shown in red, as well as DAPI for the nucleus. And what you can see in these images is that NS1 is present in the cytoplasm. It is also present in the nucleus. So in order to better visualize NS1 in the nucleus and to concentrate it, we next treated A549 cells that were infected with RSV with the inhibitor called KPT-335. And this inhibitor binds to the exportin XPO1, also known as CREM1, which is responsible for transporting cargoes from the nucleus into the cytoplasm. And what we found is that when we treated these cells with KPT-335, that both XPO1 and NS1 is concentrated in the nucleus. They're also co-localized. Whereas if we stain for a different RSV protein, nuclear protein, this protein remains cytoplasmic. We also performed cellular fractionation studies to further characterize NS1 in the nucleus. And shown here are the results from a co-immunoprecipitation. These are the Western blots showing from the co-immunoprecipitation. And what we can see here is that NS1 is present in the cytoplasmic fraction. It is also present in the soluble nuclear fraction along with different mediator components, and is also chromatin associated along with the histone H3 marker. Now, because NS1 is associated with chromatin, we next wanted to map chromatin binding of NS1. So we next performed ChIP-seq on A549 cells that were either infected with RSV or transfected with NS1. And this was work that was done in collaboration with Jackie Payton at WashU. She performed ChIP-seq for both NS1 and for mediator and was able to identify 1,700 peaks for NS1. And of these peaks, about 40% coincide with mediator. To further map and identify which regions the NS1 binds to, she performed genome annotation analysis, which revealed that NS1 was enhanced, particularly at the promoters and enhancers, as opposed to non-coding regions of the genome. And this also, binding of NS1 also coincide with genes that were altered or upregulated during RSV infection. So we next wanted to ask whether NS1 binding to these regions would decrease transcriptional activity driven by these NS1 binding regions. So we next wanted to focus at the IFIT locus, where we saw particular enrichment of NS1 peaks, 3 of which here overlap with mediator. So each of these NS1 binding regions was then cloned into a luciferase reporter and transfected either with NS1 or an empty vector. And what we found was that NS1 did decrease transcriptional activity that's driven by 3 of 4 of these NS1 binding regions. And these results suggested to us that NS1 preferentially bound to regulatory elements that were upregulated during RSV infection. So we next wanted to ask whether the C-terminus of NS1 is important for modulating host gene expression. The heat map on the left shows expression of genes upon wild type infection, and what you can see that there's a number there are a number of genes that are significantly upregulated, and these same genes are significantly downregulated when we introduce a mutation in the C-terminal helix of NS1. And this is also reflected in the volcano plots in the middle of the slide. And so given this difference in expression, we thought perhaps the mutation in C-terminal helix impacted the ability of NS1 to localize into the nucleus. However, we found that the NS1 mutant was also present in the nucleus at levels that were comparable to that of wild type NS1. So to test if mutation in the C-terminal helix impacts NS1 suppression of transcription driven by these NS1 binding regions, we next express a luciferase reporter containing the interferon stimulated response element, and we find that wild type NS1 can potently inhibit transcriptional activity of this ISRE, and that mutation in the C-terminal helix results in a partial abrogation of the suppression. And so altogether, this data suggested to us that NS1 may interact with 1 or more transcription factors to modulate the transcription of host immune response genes. And so finally, to summarize, we were able to solve the X-ray crystal structure of NS1, which revealed that it is a structural paralog of RSV matrix protein. We identified a region in NS1 that was important for its interaction, the C-terminal helix, which includes interferon antagonism. We also showed that NS1 is present in the cytoplasm, but it's also present in the nucleus during RSV infection, where it can modulate host gene transcription. And so with that, I'd like to thank everyone in my lab, past and present, that contributed to this work. I'd like to acknowledge all my collaborators on this work as well as support from the funding agencies. And thank you for listening.